All right, thank you for everybody who's joined in to the Zoom meeting for the seminar, and a big welcome to Dr. Anna Hargreaves, who is giving us a seminar today. She is currently an assistant professor at McGill University and has a Trent connection. She did her undergraduate at Trent and then moved on to the University of Calgary after that for her master's, and then after that did a PhD at Queen's University. And I think with this sort of short introduction, just welcome Dr. Hargreaves, and we look forward to your talk.
Thank you. Yeah, it's a real pleasure to be at Trent, even if it's virtual. So Trent had a outsized impact on my beginning career as a biologist and as a researcher. It's where I had my first exposure to research and first caught the research bug. And when I was at Trent, the graduate program was just starting. Like when I began, there was no graduate program in biology, and the first students arrived by the end of my undergrad. And yeah, it was really a wonderful training in biology. 4 of the people in my itty bitty biology cohort went on to be professors of biology. So yeah, I have really fond memories and associations of Trent. It is also where I first became interested in how organisms interact with each other. And so I wanted to give a little shout out to my second year botany professor, who really sparked this interest in a lab where she brought in a bunch of flowers and asked us to look at their characteristics and use those characteristics to guess the animals that pollinated them. And this idea that that 2 totally unrelated groups of organisms were so influential on each other's ecology and evolution that you could guess who a flower interacted with just by the shape and characteristics of that flower, that I just found that so intriguing, and that that really was the spark of my interest in species interactions. And then the other side of that is that plants, I think lots of people think of plants as just habitat, like the more boring side of the biotic world, but they don't move quickly, but they have these incredible wily strategies to manipulate animals to do what they want. In this case, to carry their sperm equivalents around like little taxis. This is a really great illustration of how interesting biotic interactions are. One of the species she brought in was this Strelitzia flower from South Africa. And this flower is unlike anything we have in Canada, unlike anything I was used to thinking about, and ends up being pollinated by a combination of birds and lizards. So it also got sort of introduced me to this idea that interactions are really interesting, but they're also really variable, and you get totally different groups of organisms interacting in different places. So from my undergrad, I went on to do my MSc, and at this point, I had the pollination bug pretty badly. And so I did my whole master's degree in South Africa looking at pollination systems of aloes. And for any of you who are grad students, I think it's always reassuring to know that several things went dramatically wrong during my MSc. One of them was that we had a fire move through one of our sites and burn down an entire set of experiments that took 2 weeks to set up. But another thing that that happened was that I was doing a 4-site comparison, and 3 sites behaved the same way. We got the same results, and 1 site did something totally different. And we had these 4 sites as intended as replicates to add strength to our arguments that we were finding general patterns. And so at the time, it was really annoying that this one rogue site was doing something totally different. But as I was thinking about the results for my MSc, I became really interested in the idea of spatial variation as something that should not just be like an annoying thing that you hope doesn't exist in your experiment, but something that's really interesting in and of itself. And so how do these biotic interactions vary across space? So that's my little shout out to Trent's influence on my whole research career. So we know that interactions between species can be really powerful ecological and evolutionary forces. We know that they can drive population dynamics and population cycles. This is an example of snowshoe hares and lynx that comes from Canada. We know that they can strongly affect individual fitness. And by affecting individual fitness, they can select on the traits that are involved in interactions. So this example is from South Africa, and it's this beautiful study where they looked at geographic variation in the length of the proboscis of these long-tongued flies and the length of the flower that they pollinate. And so what they found was that when you get spatial variation in a biotic interaction, so for example, in the tongue length of flies, you can get spatial variation in the traits that are involved in interactions such that organisms are locally adapting to their biotic community, their local biotic environment. We know that this type of local adaptation to biotic interactions can actually scale up to speciation. So probably the world's most famous example of speciation is the Galapagos finches. And this is really speciation driven by a biotic interaction, so driven by the interaction they have with their seed prey. And finally, we know that they can scale up in another way to affect species distribution. So this is an example that comes from Quebec, where seed predation was found to be the single most important factor that influenced the distribution of sugar maples. So there's no question that biotic interactions are really important. They can be really important for ecology and evolution. But incorporating interactions into a systematic understanding of spatial patterns in ecology and evolution has been tricky. And so to illustrate this, imagine that you are studying some sort of spatial process and you want to look at how abiotic factors influence it. You can download incredible data on abiotic factors such as climate in a matter of minutes. And this data will be globally comprehensive. A whole bunch of people will put a lot of time into truthing it and accounting for various factors. And these data are easily available. And part of the reason is because we have really standardized ways to measure abiotic features. So we measure them in standardized units. We have a standardized approach. We put out weather stations. We collect climate data. And there's really no equivalent for measuring biotic interactions in a systematic way. In fact, the best that we've done so far really is to look at the distribution of individual species. And so this is the best, most beautiful map of biodiversity currently available, and it only deals with vertebrates. So, we would be pretty excited if we could even just map where species were, let alone how they interact. And this lack of data leaves us in this situation where we have lots of really compelling case studies that show that interactions can be important, but we have much less predictive ability about where we expect them to be important and where we need to go out and collect the data to incorporate it into our predictions and models. So this has led me to the question of, are there geographic patterns in the importance of species interactions, predictable spatial patterns? And like any good question in ecology and evolution, people have thought about this long before me. One famous example is Dobzhansky. So he's best known for his evolutionary work on fruit flies, but he also wrote this really influential paper called Evolution in the Tropics. And at the very end of the paper, he proposes that interactions between species are more intense toward low latitudes, so towards the tropics. And because of this, he proposes that they're more evolutionarily important in the tropics, so more important for driving adaptation and eventually even speciation. In a really similar vein, but going back even further, we have a prediction from everyone's favorite hypothesis generator, Charles Darwin. In The Origin of the Species, he proposes that species interactions are not only more intense towards low latitudes, but also more intense towards low elevations. And that because of this, he proposes that they more often limit the low latitude and low elevation edge of a species distributions. So we have these conjectures about the importance, spatial importance of biotic interactions, But until recently, we haven't had data at the scale needed to really test these predictions. So I'm going to tell you about essentially one key result from 3 totally unrelated studies that look at this question: are there geographic patterns in the importance of species interactions? Each of these studies is its own data set, its own methods, and in each of these cases, this is just one of the questions that these studies asked. But they're interesting because they all relate to this central question. So from an ecological perspective, we tested whether interactions actually are more important or stronger at low elevations and low latitudes. From an evolutionary perspective, we asked whether how often interactions influence local adaptation, and tested Dobzhansky's prediction that they should do so more often in the tropics than in the temperate zone. And finally, we looked at how often interactions limit species biogeographic distributions, and we tested Darwin's conjecture that they should do this more often at the warm, so low elevation and low latitude edges of species ranges. So we will start with this first first question. So in this case, this was a standardized experiment, which means that I and my collaborators actually went out and collected the data in the field ourselves. And so what's cool about a standardized experiment is that you do the same experiment in a bunch of different places using standardized protocols and standardized materials. And this gives you really the combined power of an experimental approach with a huge geographic scope that you don't usually get from experiments. So the interaction that we used was seed predation. Seed predation is a great interaction for this study for 2 reasons. First of all, it's really ecologically important. So seeds are the babies of the plant world. They are vulnerable. They are really important. They are delicious. And different patterns in seed predation can have lasting effects on plant communities. And then, of course, as I told you before, seed predation can, in fact, structure geographic ranges in some cases. The other great reason for using seeds is that they're stable. So you can take seeds and you can ship them somewhere else and they don't change, which means that we were able to use the same 2 agricultural seed species throughout our entire experiment. So the experiment that we did was really simple. So we would go to a site and we would set out little depots, like just make a little depression in the ground and put in a few seeds. Then we would set out 30 depots of one of these 2 species, sunflower seeds or oats, at a site, and then we would come back in 24 hours to see how many had been eaten. It's a really simple experiment. The trick was to replicate the experiment at the geographic scale that's relevant to this prediction. So each of the triangles that you see along the west coast of the Americas, each of these is not just a site where we did this experiment, but a mountain where we did the experiment in a transect of sites spanning usually about 1,000 meters of elevation. And that means that we can look at both elevational patterns and latitudinal patterns, and we can tease them apart. This experiment, it's not the kind of experiment you do by yourself, much as traveling would have been really nice. And I worked with a team of really just wonderful collaborators from 7 countries and many invaluable field assistants to collect this data. And so each collaborator was in charge of a different transect, and they ran the experiment multiple times, in most cases, usually 4, over a series of 2 or 3 years. So the first result that I want to show you is the variation that we had in our results. So each of these lines shows the elevational pattern at one of those 18 mountains. And you can see that we have all possible patterns happening in our data. And the take-home message from this slide is that you can't test a global hypothesis at a local scale. because there's too much variation. So if you tested this at any one of these mountains, you could have found any possible result. You really need to be asking big geographic questions at big geographic scales. And because of the scale of the experiment that we did, we could pierce through this variation and get some really clear, consistent results. So we found that there was, no matter how you slice the data, seed predation was higher in the tropics, so at low latitudes, and declined towards the Arctic, high latitudes. This pattern was consistent whether we looked at total seed predation, so seeds were open to everybody who wanted to eat them, or whether we used cages to exclude vertebrate predators and looked just at invertebrate predation. So that's this gray line here. I'm not showing you the result, but we found the same pattern for elevation, that seed predation was consistently higher at low elevations and declined towards high elevations. In our study, we found the effect size was about 18%. So there's an 18% increase in seed predation from the Arctic to the equator. In fact, I think that the true effect sizes are quite a bit higher than that. I've just been digging into these data again and analyzing them in a different way. So this is a big effect. This is in 24 hours, the first 24 hours of your life, you have a much higher chance of getting eaten in the tropics and low elevations. At the time that we did this experiment, it was the most ambitious test of this conjecture. The interactions were strongest in the tropics and lowlands, and we find robust support for that hypothesis. Other studies have also been testing this hypothesis, and the biggest studies with the best standardized methods are finding results really similar to ours. So consistent increase in the importance of interactions, particularly predation, which is by far the best tested interaction, towards the tropics and lowlands. So the next study that I wanted to tell you about was the second one, looking at evolution and the patterns in local adaptation. So for this study, we took a different approach. We did a meta-analysis. And again, I was lucky to work with a really excellent group of collaborators. Megan, Rachel, and Amy are at UBC. Josh was in my lab at McGill at the time that we did this. And I'll just start by refreshing your memory about how local adaptation works. So local adaptation is the idea that if you have an environment that varies in space, so for example, here we have variation from green to purple, under the right conditions, local populations will adapt to increase their performance in their local environment. And the telltale sign of local adaptation is if you then move individuals from different populations to a common environment, you expect that the local genotype will outperform the foreign genotype. So in this case, this blue plant from the blue environment has higher performance than these foreign populations in the blue environment. And that's what we call local adaptation. So this type of experiment, which is called a common garden experiment, where you move things to a common environment, has been done for a long time. We have more than a century of these experiments. But everybody does them in slightly different ways. So one of the big differences is the type of environment that you use. So one approach is to leave natural interactions intact. So you would go and you would have your common environment in a totally natural setting. And these studies are really testing for local adaptation to the full suite of abiotic and biotic factors in the environment. But it's also really common for researchers to alter the environment that they're doing their study in. So for example, imagine I was going to do this experiment with seeds. I might take my common garden and clear out all the natural vegetation before I plant the seeds. And essentially all of these ameliorations, what they're doing is they're getting rid of negative biotic interactions that could reduce the overall success of their transplants. So in that example I gave you, I was getting rid of competition. And so if biotic interactions are commonly driving local adaptation, then we expect that studies that are done in a natural environment should be detecting a stronger signal of local adaptation than studies that are done in where they reduce the biotic interactions their transplants are exposed to. So what we did is we compared the results from this group of studies where interactions were intact to results from this group of studies that had removed some potentially important interactions. We looked at different measures of local adaptation. So we looked at the strength of local adaptation measured in 2 different ways, and we looked at the frequency of local adaptation. I'm just going to today tell you about the frequency results. So essentially, we said that local adaptation is detected if if local individuals outperform foreign ones. So to test Dobzhansky's prediction, we split the data in one of our analyses into temperate studies and tropical studies. So I'm going to show you the temperate results first, partly because they make up the vast majority of our data. So more than 90% of the data in our analysis was coming from temperate environments. We replicated our search in Spanish. We did specific searches targeted at tropical environments, and this imbalance persisted. So we just have way more data from temperate ecosystems. So here we're looking at the probability of local adaptation. There's a line here for your reference at 0.5. So if we're here, that means that local adaptation is being found like 50% of the time, which is no different than chance. And we're starting with the experiments that leave interactions intact. And what we find is that temperate systems, we detect a strong overall signal of local adaptation. So we're detecting local adaptation significantly more than 50% of the time. These boxes show 95% confidence intervals, and the dots here show partial residuals from a binomial model. So then we look at this other group of studies that's removing some potentially important interactions, and what we're expecting is that they should be finding local adaptation less often if biotic interactions are actually important in driving local adaptation. And what we found totally did not meet our expectations at all. So we find no indication that studies in natural environments are finding local adaptation more often than studies that are removing biotic interactions, and therefore no indication that in temperate environments, biotic interactions are routinely driving local adaptation. If you can't think of anything else to ask me about in the question period, ask me about this. This is a really odd result. We were totally not expecting it. I think all of us are, from our own field experience, have seen the importance of biotic interactions in the field and expected that importance would translate to a role in driving local adaptation. And the crazy thing is that even in our data, some of our data enabled us to directly test the fitness effect of biotic interactions. And we find that in temperate environments, biotic interactions have a strong fitness effect, but they're still not driving local adaptation overall. So this is our result from the temperate zone, but as I promised, we'll also look at the tropics. Because in our data, there is a significant interaction between the effect of removing biotic interactions, so the effect of this difference, and the latitudinal zone in which the experiment is conducted. So as I mentioned already, we do not have a ton of tropical data. So we're not in a great position to make really definitive statements about how interactions are affecting local adaptation, or even just about local adaptation in the tropics. This is a big data gap that, that we as scientists could and should fill. But the results from the data we do have are much more the pattern that we were expecting. So studies that are doing their study with all interactions intact are finding a stronger signal of local adaptation than those that are removing interactions. the take-home message from this, aside from the fact that we need much more tropical data and that something weird's happening with temperate systems, is that local adaptation seems to interactions seems to be more common in the tropics, as predicted. So this brings me to the third study that I wanted to tell you about. I'll just give you a pause to digest that, and I will have some water. Which is looking at Darwin's prediction that biotic interactions should be systematically more important in limiting the warm edges of species distributions. So this study, I worked with an undergraduate student in my lab, Alex Paquette, and we did a systematic review of the causes of species range limits, where we scoured the literature for studies that had evaluated any potential driver, abiotic or biotic, at either a cool or a warm range limit. And the motivation for this study actually came from 2014. So in 2014, we published a review of transplant experiments that had moved species beyond their range limit. And one of the things we tested in this study was whether biotic interactions were identified as limiting the range more often at warm range limits. And what we found was that the results support the importance of biotic interactions at range limits in general. So they are important in driving range limits, particularly the long-held assertion of their role in causing low elevation and equatorial limits. So in our study, we find that biotic interactions are more important at species warm versus cool range limits. In the very same year, another synthesis was published in the Journal of Biogeography, and they had a different approach. So they were looking just at warm edges, whereas we were comparing warm and cool, and they used different data. So in addition to transplant experiments, they looked at observational studies and models and lab experiments, and they reached the opposite conclusion. So they found that abiotic factors are supported more often than biotic factors in setting species range limits at warm edges, in contrast to the widely held view that biotic factors are more important. And so this contrast in results has always niggled at me, and I've always wondered how it was that these two studies arrived at such different conclusions. Whether we were testing different predictions, which we were testing slightly different predictions, or whether it was the different data, or what it was. So finally, Alex and I set out to solve the mystery. And so we surveyed the literature for studies of cool limits. So this is this could be a high elevation limit. It could be a high latitude limit, terrestrial. It could be a high latitude limit in marine systems, and warm limits. So again, low elevation or low latitude. And we found a solid number of studies, so well over 300 studies, with almost 1,000 tests of cool limits and warm range limits. So where each test is one species times one range limit. So that said, the data were relatively evenly divided between cool and warm limits, but they were unevenly divided between biotic and abiotic factors. So we found almost 3 times as many assessments of abiotic factors. So the blue dots here show the assessments at cool limits. The red dots show assessments at warm limits, and not quite 500 tests of biotic factors. And so, so what this reveals is that we are looking far more often at the influence of abiotic factors on species ranges. So even though as a field, biogeographers and ecologists are increasingly realizing that biotic interactions can limit species ranges, we're not assessing their importance nearly as often as we assess the importance of abiotic factors. So, so this is just the distribution of the data. Today, I am just going to tell you about the results for biotic factors. So here we're looking at the y-axis is the probability that biotic factors impose a given species range edge. And we've divided the data between cool range edges and warm range edges. And this data is across studies. So we've included here field experiments, lab experiments, observational studies, so field data but no experiment, and models. Models usually often are SDMs or other cases where people have just scraped data from globally available sources. And what we're expecting, according to Darwin's conjecture, is that the probability that biotic factors impose a range limit should be lower for cool range limits and higher for warm range limits. Unlike the last study I showed you, we actually find the pattern that we expected. Again, the difference, no matter how you slice it for this particular result, the difference is significant. So biotic factors are significantly more important at species warm limits than at species cool limits. And at species warm limits, they're important more than 50% of the time that they are assessed. So I am a field biologist and an empiricist at heart, or an experimental biologist at heart. And so these results are cool, but they bother me a little bit in that, we've done this study replicating the methods by Cahill and all, but 3 of the 4 methods that are included rely really heavily on correlation to establish causation. So for example, models can look at a correlation between where a range limit is and some other factor, but they don't have any ability to test whether that factor is really causing the limit or it's just correlated to something else that's causing the limit. And because we have way better data on abiotic factors, models are generally testing abiotic factors, which of course could be correlated with biotic interactions instead of having a direct role. So we redid this analysis using just field experiments. So these are experiments where people manipulated something in nature. And so they vary also in how good they are, but they're at least a little bit better able to establish causation. And we find the same result, only much stronger. So again, biotic interactions are more important in limiting the warm edges of species ranges than the cool edges of species ranges. And biotic factors are supported significantly more often than expected by chance at warm range edges. So these are the 3 results from 3 totally independent studies, each of them looking at a different prediction about the geographic patterns and the importance of species interactions. And we find that interactions, in this case specifically seed predation, are indeed strongest in the tropics and lowlands. We find local adaptation to interactions has some interesting and unexpected stories, but it does seem to be more common in tropical ecosystems than temperate ecosystems, with the caveat that we definitely need more tropical data. So supporting Dobzhansky's prediction, and interactions are most important at limiting species warm range edges, supporting Darwin's prediction. So the answer to this question, I think, is to the best of our ability to tell right now, the answer is yes. There do seem to be systematic patterns in the importance of species interactions. And so this matters for a few reasons. From a fundamental ecology perspective, one of the big questions in ecology is which ecological factors drive ecology and evolution, and whether we have any predictive power. This is an ongoing debate in ecology. How much predictive power do we have? Can we predict where different factors matter? And so so the answer seems to be, with lots of variation, as one expects in biology, yeah, you can start to make at least first hypotheses about where interactions are most important just based on geography. But these results are also important for conservation, and I'll just give you 2 examples. One thing they suggest is that we should expect stronger effects of species loss in the tropics. So if interactions between species are more important in driving ecology and range limits and evolution in the tropics, then taking species out of those systems we should expect that to have direct impacts on other species. A second example is our ability to predict what's going to happen with climate change. So this is tons of people have spent tons of energy and still are trying to imagine or predict how species are going to move in response to climate change. And our results suggest that warm limits should respond less predictably to changes in the abiotic environment than cool limits, because they are more influenced by interactions, and the more species influence range limits, species are all responding idiosyncratically to climate change, and that noise should be more apparent in the response of warm range limits. So uncharacteristically for me, my talk was slightly short, and this is the first time that's ever happened. So I do want to end with just a shout out to some students in my lab who are working on really related questions. So Antonio is looking at new data we have from that global seed predation experiment. Ella is also looking, doing a synthesis on seed predation. Emma was looking at global patterns in pollen limitation. And Shannon's been doing this epic experiment manipulating parasitism along elevational gradients. So if you're interested in these questions, you can check out their work on my webpage. And with that, I'm totally happy to take questions. Thanks for your attention.
Thank you very much for your talk, Dr. Hargreaves. So we will open up for questions. Everyone feel free to unmute yourself and ask, or if you write in the chat, then I can help moderate the question period.
I have a question.
Go ahead.
Thanks, Anna, for that.
Hi, Erica.
Thank you. Really nice to see you.
You too.
Yeah, great talk. My question was, well, I have a couple questions, but I'll just ask the one in the last study where you showed a strong effect of the interactions in the tropics at the warm edge of the range. What were the main biotic factors? You had mentioned predation for the seed experiment, but I'm curious.
Oh, yeah. I should have put an emergency slide for that question. The single most important factor at warm limits was actually biogenic habitat. And so that is habitat that's created by other living organisms, usually photosynthesizers. And what's interesting there is that in all my years of reading people debate about these hypotheses, I don't think I've ever seen anyone raise biogenic habitat as the potential interaction that drives warm range limits. And it's not clear theoretically why it should, but it was significantly, even by itself, it was like when you do the parse out the interactions, it's a different analysis. It's significantly more important at warm limits than cool limits. Another interaction was competition. So competition is much more like the classic interaction that people think of. And it was significantly more important at warm versus cool limits. There's another one, predation herbivory was the other one, which is also interesting because we tested a lot in standardized experiments, but it's not usually the go-to interaction people think of for the Darwin's conjecture. But the other thing, since you asked, I'm just going to tell a little bit more information than you technically asked for, which one of the cool things we found is that temperature actually is more important at cool limits than warm limits, but it's really important at both. So it's supported, it has really high level support at both types of limits. That's a bit of a tricky one to interpret because temperature is the easiest thing to get data on, and it's correlated with a million different things. And so it's, but we still find that result even in field experiments.
Sorry, can I just have a clarification? So when you say biogenic, so do you mean like an insect will disappear because the plant it's feeding on is gone?
Yeah, that's a great example. Yeah, or like or a bigger habitat requirement in general. Like this species stops occurring because the rainforest drops out here, or like you get a transition from conifer forest to prairies. Yeah, yeah.
Influenced by something else.
Oh, totally. I mean, the biogenic habitat one's so interesting because it clearly that often is like at a big scale limited by temperature and precipitation. And I think that's one of the interesting things about these ideas is that I don't think anyone would debate that climate is often setting the stage for species distributions. And so the question is, it's not really like, does climate matter, but can we predict things based on climate alone? Or are interactions having a big enough effect that our climate-based predictions will be wrong?
Hi, Anna. Nice. Thanks for the talk, Anna. It's great to see some of this more recent work from your lab. I'll ask the question you wanted people to ask you and just have you comment on the idea that biotic local adaptation or local adaptation that's influenced by biotic interactions can cut both ways. And that a plant that might be, if you remove the herbivores in a population, those herbivores might also be locally adapted to the plants that they're feeding on. It might, it could cut both ways is the point. I just wanted you to comment on that. It's probably not what's driving the pattern that you're seeing, but maybe just.
Yeah, I don't think it could exacerbate the pattern. So the idea would be you do your common garden transplant, and if you remove interactions, well, you tell me if this is what you're thinking of. If you remove interactions, so say competition is super important, you have a plant that's adapted to competition and all of the competitors are adapted to that plant or that genotype, then if you remove competition, then you lose your signal of interaction for that plant. But also if you move in foreign genotypes that the competitors aren't adapted to, they should get a little fitness boost because the community is not expecting them, so that you could get not only a loss of local adaptation from your focal plant, but a small boost in fitness for the foreign genotypes. Is that what you're thinking?
Sure.
So yeah, so that's not something I thought about before, but you're right that could further dampen the signal of local adaptation if you remove the interactions. Yeah, I mean, in terms of this result, like the weird result from the temperate zone of why we don't get a signal. So we get a signal that interactions affect fitness, but not that they drive local adaptation overall. And I should say that we definitely have studies in which they do seem to be important for adaptation, but it's just not a globally important effect. I think that, well, there's a few possible explanations. One is that in the temperate zone, interactions are too variable to bother locally adapting to. So, so essentially, you get, populations of insects go through big cycles, population of herbivores move around, competitors, might have boom and bust cycles of their own. And you only expect local adaptation when the environment is predictable. And if the environment's not predictable, you should expect phenotypic plasticity. So if temperate environments were less predictable than tropical environments, then you could have biotic interactions strongly affecting fitness, but not driving local adaptation. The other option is that we're doing these experiments in a biased way. Yeah, which I don't want to like drone on too long about this, but I think I would definitely not exclude human influence on these results.
Temperate environments are usually, the species there are usually younger too. So does that
Yeah,
make
that's true.
It depends on where they were done. The experiments were done.
Yeah, I mean, the temperate, they come from all over the show, but, mostly, I mean, as was the case with any meta-analysis, you get this band, like continental Europe, especially Northern Europe and the US, especially, and like southern border of Canada, northern 2/3 of the US. That's where so much data comes from. Yeah.
Anna, can I ask you about extreme events? How important do you think extreme events are, climatic extreme events? Because I'm thinking of things like on the prairies, there's a sort of march of the trees out onto the prairies on a regular basis. And then the occasional fire beats them back. And at climatic extremes, you often get, 50-year events or whatever. And these have a profound effect on the range of the particular species. I wondered how you thought that fitted into your general ideas.
Yeah, nice to see you, Tom. That's a great question. So in other work that I've done, we've put a lot more brain power into thinking about how you would detect the signal of strong climate events. So, if you imagine you have like a range limit, we'll just use elevation for ease. And, 9 years out of 10, a plant can survive up here, but every 10th year, there's a really hard winter and the range gets pushed back. At that point, dispersal starts to become really important. So if plants have really good dispersal or whatever, I just use plants because I always think about plants, but then they can regain that lost space quickly. And in the majority of times that you do the study, you will find that the species is at equilibrium with climate. And but if they have low dispersal, so if the magnitude of the environmental effect is bigger than the dispersal ability, they may never catch up to their, so 9 years out of 10, they could occur here, but they'll never get there because they just never have time to disperse that far up. And so that means that 9 years out of 10, when you do a study, you will find that the species is dispersal limited, and so could, in theory, live in this area above its current range, even though the actual range on a longer time scale, a time scale bigger than the average master's or PhD experiment, is probably in equilibrium with climate. So there is, at the same time, you can have weirdly favorable years that allow species to, persist beyond where they normally occur. And so I think it's a real interesting way that dispersal interacts with climate fluctuations. In our data, we generally assume that the unusual bad years and the unusual good years even out. And so that you might get the wrong answer in a single experiment, but when you look across 100 experiments, you get the right answer. Yeah, but extreme events are hard. It's hard to tease about their role, and it's an interaction between extreme events and dispersal.
Yes, it certainly is. The frequency of these events and the repeat of them is very difficult to take into account. But I'm thinking, you were saying outside of the PhD cycle and so on, which I fully understand, but we've got some responses going on still to the last glaciation. Sort of the march of beech trees going north hasn't got where it should be. It's lagging all the time. And they're making their way slowly north. And earthworms are a similar situation. They're not where they should be climatically because they're held back by migratory forces of dispersal and so on. So yeah, it's an interesting question. It's probably the sorts of things I'm talking about are probably outside of what you're actually trying to study.
Yeah, I mean, the tree migration thing's really interesting because I always think of trees as being like chronically dispersal limited, but there are studies that find that they're not. And it's not a literature I've kept up with as well as I should have, but I think there is some counter evidence from some places. But yeah, I mean, and then the other thing that's interesting about these types of studies is that whenever you're looking for matches between organisms and their climate environment, more and more we have to take into account the fact that the climate environment is no longer the ones that these organisms evolved to. Yeah, so I was involved, this same data set that's shown in this slide, one of the other studies that we've done with it, not that's spearheaded by me, that's spearheaded out of Amy Engert's lab in UBC, has been looking to see whether essentially we detect smaller and like less and less reliable signals of local adaptation as time goes on. So the more recent the study of local adaptation to climate is, the more likely you are to get a signal of maladaptation to that current environment. And then it's a really complicated question. The analyses are like, yeah, they're intense, but essentially there is a signal that that the effect of climate variability is there in the data, and that the signal of local adaptation is degrading essentially.
Yeah. Okay, thank you.
Yeah, my pleasure.
Hi, Anna. It's nice to see you here.
This is like a reunion, guys. Tom was my very first NSERC research supervisor, and Erica supervised my honors thesis, and Sandra was my first student in my lab at McGill. Like totally wonderful.
Yeah, so my question was actually also about this slide. You mentioned that you had less data for the tropics and that you even did a search in Spanish. So I was wondering if you thought that there were geographical, regional effects also on the question that you were testing. So for example, I don't know if you had any data from Africa, but do you think that the tropics there would have a different signal than the tropics in South America?
Yeah, I mean, that's a really good question. And it's one of those times where I, like, now that I look at it, I kick myself for not also doing the search in French, which we totally could have done. My sense, I mean, the honest truth is I don't, even from Latin America, like even if we could all, I think there is one tropical Africa study in here, but there might not even be. It might be all Latin America. But even if we had the same number of studies from tropical Africa, it wouldn't be quite enough to test the difference. But it's, I mean, yeah, it's super interesting. Like the biogeography of Africa, it's so different than the Americas because, you know, if you think of the Americas, you get a real pinch point around the tropics. Like there's very little land there in Central America, and that's not true of Africa. Like the band of the tropics in Africa is huge, and then land narrows down to the temperate zone in the south. And then you also have the effect of the Sahara, which is like, can really, you know, so in our data, we say, we just lump all high latitude limits together and low latitude limits together, but there's a big chunk of Africa where that pattern is reversed, where like the limit that's towards the Sahara Desert is much more abiotically demanding than the tropical limit. And so we haven't, yeah, we rarely have the data to tease the effect apart. We have much more data actually for the literature survey, partly because we did that search in French and Spanish. But also because the criteria for the local adaptation study were quite restrictive. Like, you know, you had to do a common environment experiment, you had to use a local genotype within X kilometers, you had to measure some component of fitness. So like we didn't count studies if they just measured growth. And so we ended up discarding quite a few really cool studies that just hadn't done the experiment in the way that we'd set up our parameters to analyze. And the survey, the synthesis, it's less powerful because it's not a meta-analysis, but it's a much bigger catch net. And so we can look in a little more detail. But I haven't tried yet to, I've been thinking about trying to disentangle the effects of the Sahara specifically, but I haven't done it yet. Yeah, thanks, Sandra.
There's one question in the chat from Dr. Xenopoulos. She says, great talk, but she just wanted to know, who is the Trent botany prof who inspired you?
I texted Erica this morning because I couldn't remember her name. Erica or Tom?
Christine Maxwell.
Thank you, Christine Maxwell. Yeah, she retired a while ago now.
Yes, quite a while ago.
Yeah,
Sorry,
yeah, she was lovely. She actually, one of her assignments in her class, I can't remember what the assignment was, but we ended up doing a spoof video of a David Attenborough documentary where we like juggled fruit, and she mailed a VHS tape, this I'm dating myself, a VHS tape of our assignment to David Attenborough because she thought he would enjoy it. We never heard back. I'm gonna write down her name so I can give her a proper shout out next time.
Well, we have time for one or two more questions.
I have a quick question. I'm not sure, maybe you explained this and I just missed it, but could you explain like why, in the first place, like why Darwin and Dobzhansky actually predicted that interactions would be more important in the tropics?
You didn't miss it, and that's a great question. The truth is they're really vague about the mechanisms, and it's something that has been frustrating when one tries to test these predictions, because the mechanism is not clearly laid out, and therefore the definitive test is not always clear. So we've kind of been left to piece together the mechanisms sort of post hoc, like on our own. And there's a few, so in the Science Advances one where we tested the seed predation gradient, we lay these out, and actually we test support for them. But one idea is that it's just totally temperature driven. So if you have a warmer environment, especially things like cool-blooded organisms can have higher activity levels. And so you can just get, for example, higher seed predation because you have the same number of animals, but they're all eating more because their metabolisms are amped up. Another way that it could happen is actually through habitat diversity, often driven by like net primary productivity. So the more plant biomass you have, the more structure you get to the environment. And that means that you can get what's called niche packing, where you have different niches that organisms can live in, and they can then exploit slightly different habitats, and that can support a higher total number of species. So if you have a higher total number of species and each one has a similar number of animals, you can get more animals in an environment doing an interaction. So for example, for seed predation, you can increase it by having each animal is more active, or you have more seed predators. And then there's often, this hypothesis is often linked to just like raw biodiversity. And it's like an intuitive link where you feel like, oh yeah, if you had lots and lots of things, then you would have way more pairwise interactions. But when you really try and like nail it down, it's not quite as clear as that. So the easiest route from high biodiversity to more intense interactions is if it also allows you to have more niche packing. So you can fit more species in an area, and that increases the number of actual animals or individuals in the area. But it's a great question because it's very intuitive, but I think people make the prediction based on what they're seeing, and haven't always like been really good about spelling out the mechanism.
I think we have one more question from Susan.
Hi, Anna. I was just wondering, since the system's not static and there's constantly changing, how do these organisms manage to actually achieve this local adaptation like so quickly? And also, do you know if there is a difference in terms of like changes in temperate versus say tropical system, like the speed of changes?
Speed of adaptation.
Yeah.
Oh, that's a cool question. I mean, the easy answer to that question is no, we don't know, because we don't have that component of data in our study. You could get at it maybe, I'm just thinking whether you could even get at it in our data if you had a phylogenetic tree and you looked at sort of branch length, like how old the species were. We have the species identity of the focal species in the experiments, but often the interactions that are affected are general. So like the study will say we removed all the vegetation, or we caged the plants to exclude herbivores, or we did, you used pots, which means they don't have their normal microbial communities. But we don't know the identity of those interactions. So at least from our data, we wouldn't be able to tease that apart. I mean, there's a whole industry of people who study whether speciation, like, is tropical diversity higher because species evolve faster, because we lose species less quickly. I am like not good at keeping up with that particular literature. But yeah, but there's some evidence that species actually speciate faster in the temperate zone. So I don't know whether we could guess at the age of the species. In terms of like how local adaptation happens quickly, it really depends on the strength of the selection and the amount of gene flow you have. So so one thing that's, a classic example of local adaptation in plants, at least, is to soil. So you can have local adaptation to serpentine soils, which are high in metals, but you can also have adaptation to tailings, also high in heavy metals. And they're part of what happens is you're just getting constant dispersal from the surrounding area because the soil is quite localized. So you have a lot of immigration, but then really strong selection once you get there. And so those kind of conditions can yield local adaptation relatively quickly, like in as little as 10 generations in some cases. But it's this whole like melange of, how much standing genetic diversity did you have to begin with? Yeah, it's a cool question, but generally we don't have the data to like delve into that.
Thank you.
Yeah, my pleasure.
All right, well, we will end the questioning period there. And once again, thank you so much, Dr. Hargreaves, for coming in and giving this excellent seminar. And thank you everyone for a really great discussion.